Hello, everyone. Um, Welcome to Everest webinar. We're going to give it a couple of minutes for everyone to attend, so we'll start at 11 sharp. Hi, everyone. Welcome again. We're going to give it a couple more minutes to allow everyone to join, but thank you for joining.
Hi, Dima. Hello.
All right. Welcome, everyone, to the Everest Biolabs webinar series. My name is George Daboul. I'm the CEO and co-founder of Everest, and I'm glad to see so many participants here to listen to what we have to say. First, before I introduce our speaker, I would like to introduce Everest Biolabs and our mission and what we're trying to do. So Everest was started a year ago. The team of both scientists and engineers, and our mission is to solve some of the critical issues in exosome or extracellular vesicle analysis. So there's a lot of great products out there that address one of these components, but there's nothing out there that we've experienced that really solves the problem as an ecosystem. So there's a lot of issues still with reproducibility. Once you get great results, how do you scale those to look at large studies, and a really fast and easy way to analyze EVs and exosomes. So what we built over the last year and we'll continue to work on is building an ecosystem to really facilitate your research in purifying extracellular vesicles and analyzing them. So here is a little snapshot of what we have as products. We've addressed automation through instruments that we've engineered in-house to run size exclusion chromatography, either in a column format or in a plate-based format. So you're allowed to run tens to hundreds or even thousands of samples. Also, we've done some innovation on the column side of things, where we have columns that are based on saccharose resins that you're probably familiar with, but also we've just released a new multi-mode resin for really high lipoprotein purity. And also we've built analytical tools, mostly around ELISA, that depend on intact EVs that allow you to quantify EVs directly from sample or after purification with a really high sensitivity and large dynamic range. And with my pleasure, I would like to introduce our speaker, which his work kind of paved the way to developing a lot of these products. So Dima, he's a senior staff scientist at the Wyss Institute. He's part of the Wyss Diagnostics Accelerator and the Synthetic Biology platform. And he spent over the last decade perfecting EV isolation and with a focus on looking for brain-derived EVs for diagnostic applications. And with that, I'll hand over my screen and happy to give it over to Dima.
Great. Thank you, George, for the introduction and for inviting me. Can everyone see my screen? Yeah. I will go ahead and start then. So my name is Dima Ter-Ovanesyan. I'm excited to tell you about our work working on EV isolation and analysis for biomarker discovery. First, just wanted to highlight there are a lot of people that have contributed to this work, especially Tal, who was at the Wyss Institute working with me and is now head of R&D at Everest. And as George mentioned, I'm also a co-founder of Everest, and I think you'll see some of the ideas that are in this research were now distributed to a wider field by Everest in the community. So I'm going to talk about extracellular vesicles, also called by some people exosomes. I'm going to refer to them as EVs. So these are vesicles that are released by all cells into biofluids. And of course, there are many possible biomarkers in a biofluid, not just EVs. There are proteins, there are circulating nucleic acids like cell-free DNA. And we really think about EVs as not necessarily something that is better or worse than these things, but something that can actually complement some of these other biomarkers. And in particular, we're trying to figure out what are the unique features of EVs that are better for some biomarker applications than these other types of biomarkers. So that's what I'll try to focus on today. And so the experiment that got me very excited about using EVs as biomarkers now a long time ago at the beginning of my PhD was taking cells in culture and EVs and then doing RNA-seq on both of them. And one of the things that I found was that the transcriptome, this is mRNA of EVs, really reflects what's the transcriptome in cells. And what this suggests is that the EVs are a way to read out what's happening in cells, which got me very excited about using these as biomarkers because it basically means that we could read out inaccessible cells from biofluids and maybe get some of the same information as if we, in some cases, even take the cells. So this was very exciting. And so I think my work can be kind of divided into two goals. One is looking at total EVs, which is simpler. You just isolate them from a biofluid. You don't know what cell types they come from. And then a unique feature of EVs, but also one that is much more challenging, is can you take cell type specific EVs from a particular cell or organ and analyze the cargo of those. And in both cases, we're interested in both RNA cargo as well as protein cargo, and both particular RNA or protein species or more omics approaches where we can look at everything. And so the first question, if you're interested in EVs, is of course, how do you isolate them? And this is seemingly a simple question, but is actually something that I think is very challenging for many studies, both biomarker studies and also studies that use EVs to try to study their function. And that's because there's no perfect way to isolate EVs in general. But even for a given application, you can't figure out the right isolation method if you don't have a way to quantitatively measure EVs and compare EV yields across different possible isolation methods. And the reason this is difficult is because EVs are heterogeneous. There's not one RNA or protein or other component that's on all EVs. And then if we use techniques that are popular still, such as nanoparticle tracking analysis or other particle measurement methods, these work, I would say, poorly in biofluids because there are other particles that overlap in sizes with EVs, such as lipoproteins that are more abundant, as well as proteins that can aggregate into particles that, again, are more abundant and the same size. And so this makes it these techniques not very useful, in my opinion, for quantifying EVs. And then on top of that, EVs are also low abundance, so not all techniques are sensitive enough. And so our approach has been not to focus on particles, but really on proteins that are on EVs. And so for this, we turn to the standard ELISA, where you have a capture and detector antibody. But working with David DeWalt's lab, we focused on a technology developed in the Walt lab and now commercialized by Quanterix called Simoa, or digital ELISA, which is similar to ELISA in that it has a capture and detector antibody, but instead of doing it in a tube or plate, you do it in microwells where only one immunocomplex fits per well. And so then it's a digital ELISA. So in some cases, this gives us extra sensitivity, but fundamentally it's still an ELISA, and the main approach is using an ELISA-based approach versus using a particle-based approach. And so the first thing we did, which we published in 2021, using this approach, is we just compared different isolation methods using the tetraspan and Simoa immunoassays, and also an immunoassay against albumin as a measure of free protein contamination. So we compared from a given biofluid, we used plasma and CSF, different purification methods, and then compared tetraspan and albumin levels, all on the same platform. And so this was a way to quantitatively compare relative amounts of EVs, both in terms of their yield and purity. And so even though any one of these tetraspan proteins is not on every single EV, we found that by comparing these different EV proteins together, we could do a very good job quantifying relative EV yield and purity. And so we started by comparing the methods that were used at the time for biofluids, size exclusion chromatography, ultracentrifugation, kind of the old school method, and various precipitation methods like the ExoQuick PEG precipitation. And then for each sample, we started with pooled plasma or CSF, and then we measured CD9, CD63, CD81 as the tetraspanins present on EVs, and then albumin as free protein contamination. And what we found, I'm not going to go into all the details of the paper, but basically that SEC was the technique that seemed the most promising, and we were using commercial columns that were available at the time, but we wanted to use these immunoassays to actually optimize SEC because we didn't know if the commercial columns were actually optimized or whether they just took what was published at the time. And so we compared the different resins with different pore sizes, Cepheros 2b, 4b, 6b, where 2b has the largest pore size and Cepheros 6b has the smaller pore size. We compared different column heights and fraction numbers, as well as compared it to the commercial eyes-on columns. This was the first generation at the time. And for those not familiar with size exclusion chromatography, this is this technique where you have a column filled with resin, you put a sample on top, and it's fractionated based on size because the smaller things kind of zigzag through into the pores of the beads, whereas the larger things don't go into the pores and elute in earlier fractions. So this basically takes a sample and fractionates it based on larger things and smaller things, and it turns out that this worked really well for EVs. So to give you a sense of the experiments we did, here we're comparing 10 mil columns with Cepheros CL6b, 4b in the middle and 2b in the bottom, taking individual half a mil fractions and then measuring in each fraction with Simoa, CD9, CD63, CD81, and albumin. And what you can see is that in the top, the Cepheros CL6b, which had not been used at that point for EVs, actually gives higher yield of EVs than 4b and 2b. And there is a little bit more albumin contamination, but actually not a lot. And so we quantified all of this and compared it, and that's in the paper. But when we further optimized the Cepheros CL6b technique, we were able to actually get very high EV recovery of more than 50%. So the experiment I'm showing here is that we take the plasma, we measure the at the correct dilution where it's in our linear range, the tetraspanin CD9, 63 and 81 in plasma. Then we take that same plasma, run it through SEC, and then measure these tetraspanins after this Cepheros CL6b based SEC, and we get more than 50% recovery, which we thought was very good and much higher than any other SEC or non-SEC method that was available at the time. And of course, one of the disadvantages of SEC is that it's not very high throughput. Usually you run it one at a time. And so we wanted to think about whether we could run it in parallel. And with engineers at the Wyss, we built this primitive at the time device where we had a pump connected to a Raspberry Pi that just ran fluid PBS to columns in parallel. And this was just a proof of principle with a device we called the Octosome that we can run columns in parallel that could then theoretically make this relatively high throughput. Of course, 8 is not high throughput for clinical samples. And so we realized that for it to be truly high throughput, we need to move to some kind of plate-based format so that we could integrate it with automation. And we decided to use 24-well plates as opposed to, say, 96, because we still wanted to hold a lot of resin to separate large volumes of relatively large volumes of plasma or CSF. So if you want to use half a mil or a mil of plasma, you can't have a 96-well plate because it just won't have enough resin. And so we decided with 24-well plates, actually, we can hold 10 mils of resin or similar amounts. And then if we use those, we could integrate them into existing liquid handling platforms. And so we built various devices and adapters on the left. This is a device that holds the 24-well plate on the deck of an Opentrons, which doesn't have the OT2 gripper. On the right, we have a different set of stands for the TCAN Fluent, which does have a gripper and is a more advanced robot, but we can actually do this with pretty much any liquid handling robot. And when we compared the plate-based SEC to the column, even though it has a slightly different geometry, we were able to get the elution to be basically the same to a column. showing that even though the geometry is a little different, the elution is still pretty much the same, and we optimized it exactly for the parameters of the plates we had. So on the left, you have the different fractions of a plate. On the right, you have the different fractions of a column with the same resin. And so not surprisingly, if you automate this process, it then becomes a lot more reproducible. So here we compared reproducibility of a large pooled plasma sample. So this was all the same 0.5 ml sample of plasma across the wells of a plate 1 and plate 2, and then across 2 different plates measuring CD63 and CD81. And you can see that it's actually very reproducible, which again is not surprising because now this process has no manual pipetting. It's all automated. And just to give you a sense of what this looks like on the deck of a TCAN Fluent robot, you have your SEC plate, which has 24 wells. The bottom, there are frits, so each well is the equivalent of a column. And now these are 24 plasma samples on the blue rack in individual tubes. So you can see that this plasma is being loaded onto the plate. Again, this is pretty much equivalent to doing 24 columns in one go, which for anyone who's done SEC is quite difficult to do 24 columns. That really pushes the boundary of just what you can do by hand. And this robot can obviously do many of these plates because it has a gripper that can move them and a lot of deck space. So you can actually run per instrument easily hundreds and maybe even a few thousand if you really optimize everything and have it running the whole day samples, again, without manual pipetting. So now you can see the plate has been moved from being on top of a waste container where the flow through goes through during the washes and the void volume to now a collection plate, 24 well collection plate. And you can see here the bottom, the frits, where the basically the each column drips down into a collection well. And so that is the automated version. And now we're very excited about this because we think that there wasn't a good solution to get really good SEC that is also amenable to high throughput clinical samples. And now we're thinking of different applications, one of which is we're very excited about doing proteomics using mass spec of total EVs. And so mass spec on plasma is quite challenging because you have these very high abundance proteins such as albumin that are orders of magnitude more abundant than other things, so that if you just run mass spec on plasma, you just get a lot of albumin and a few other proteins. And then people try to deplete albumin, and it's quite challenging. But by isolating the EVs, you're actually depleting the albumin. And so you can get much deeper coverage of the plasma proteome. So we're very excited about this. Now with mass spec, we care a lot about purity more than yield. So actually with normal SEC, we found that the purity is not as good if you just have Cepheros CL6B because it's optimized for high yield. But we can use additional techniques either to modify the columns or afterwards. So we developed a technique based on Capticore beads, which are multimodal resin beads, where they have pores like the SEC beads, but unlike the SEC beads, they have these functional groups that when a protein goes in, it doesn't come out. And so that's exciting because now you can also do this in a tube as a slurry, so it doesn't even have to be a column, where you basically mix your sample with the Capticore multimodal resin beads, and then you remove them and get pure EVs out the end. And so with plasma, with CSF, it just works directly. With a ml of CSF, you add the slurry. With plasma, there's so much protein that we first actually have to do SEC if we want to use a ml of plasma or 0.5 ml of plasma. And then we use the slurry the same way that we would use it on CSF. And then using that, we can do mass spec. So first, to just show you the purity, this is different amounts of the Capticore beads. So CC 0.3 is how we define the ratio of protein to amount of Capticore beads. And you can see that we can get more than 100-fold purity as measured by EV to albumin ratio relative to SEC. So we're getting less yield, but a lot more purity, more than 2 orders of magnitude. And when we compare this SEC to the SEC plus this MMR slurry, as we call it, with the capture core beads, when we do mass spec from the same plasma sample, with SEC, we get under 1,000 proteins, about 700 to 800. With this SEC plus MMR slurry, we get over 3,000. So more than 4 times the proteome coverage, which is very high for plasma using mass spec. Now, we are also, of course, looking at not just albumin, but lipoproteins. And the way we approach this is the same way. We made an immunoassay using initially Simoa for lipoproteins, specifically the protein ApoB100, the main protein component of lipoproteins. And if we do SEC and take the different fractions, like I showed before with the different resins, 2B, 4B, 6B, you can see that the ApoB actually is very similar to the tetraspanins, which makes sense because the ApoB containing lipoproteins like LDL and VLDL overlap in size with the EVs. So using a different resin, we can shift a little bit the where the ApoB elutes, but it still overlaps with the tetraspanins. And if we quantify this, you can see that with cephaloCl2b, you do get more, about 2-fold more EV to ApoB ratio, but actually a lot less yield, about 5-fold less yield relative to 6B. So this isn't really that helpful for most applications. So if we want to deplete lipoproteins, just changing the pore size doesn't work. Now, of course, you could do something that's not SEC. You could do density gradient centrifugation, and then you do actually get the ApoB containing lipoproteins to separate well from the tetraspanins. So I'm showing here the different fractions of a top-loaded iodixanol OptiPrep density gradient. But of course, density gradient centrifugation is very difficult to imagine in high throughput. So some people do it for clinical samples, but the throughput is very, very limited to an ultracentrifuge, which can only do 6 samples. And then if we compare, again, the relative EV yield, you do get more purity for lipoproteins as expected, but there's also less yield. And then also you have this problem where you have the iodixanol that you usually have to get rid of. So usually people do density gradient and then SEC to get rid of the iodixanol. And so you see a pretty big drop in yield with this technique, although the purity is certainly higher both for EVs relative to ApoB100 as well as for albumin. But with the SEC, we actually aren't limited to just using Cepharo. So inspired by work from Jan van Dun in 2020, he showed that you can put other resins on the bottom of the SEC resin in the same column. So he called this dual mode chromatography, where he put a cation exchange resin to deplete ApoB100, which is quite positively charged. And we did this with our cephaloCl6b, but then also incorporated captive core beads into the bottom to get even more purity. We call this trimode chromatography, and we're still playing with lots of different combinations to optimize the depletion of both free proteins and lipoproteins for applications where you need purity. But I think with these more complicated columns, you can actually get better purity than other approaches while getting also still very high yield. And so again, the reason we're excited about chromatography, whether it's just normal SEC or SEC plus something else, is that we think that with this plate-based format, it can be high throughput in a way that density gradient or other techniques cannot be. And so we're excited about this notion of using SEC to isolate EVs. And then the other advantage of SEC is that you can also, if you want, take the later fractions, which contain the free proteins and smaller things, without splitting the sample. And that's really exciting from the perspective of trying to develop a multi-omic liquid biopsy where you can measure both EVs and other things from the same sample. So that is, I think, something that is very exciting and a unique advantage of SEC, where you can get both EVs and free proteins and analyze both. And you can imagine that analyzing both would be better for biomarker discovery and diagnostics than either one alone. And so again, that's focused on total EVs. I also want to talk a little bit about cell type specific EVs, which, as I mentioned, is more challenging, but we're actively working on. So we're really interested in neuron specific EVs, although of course people are interested in other cell types as well, but some of the challenges are the same. And the idea with any cell type specific EV isolation is that you use an immunoisolation to isolate EVs with a cell type specific protein and then measure the cargo, whether it's protein or RNA inside. So again, we're focused on neurons, but some of the challenges and things I'll talk about are very applicable to any other cell type. And so when we started this, we realized one of the things we first have to figure out is a candidate EV marker that we want to pull actually present on the EVs? Because if the answer is no, there's no point on doing an immunoisolation and then measuring the downstream protein or RNA because you're not actually pulling down those EVs. And so this is something that a lot of people have not done because it's challenging to figure out how to correctly isolate EVs and measure things on them. And so again, we turned to the same SEC protocol that we optimized. So in this case, we used CL6B because we wanted high yield. But now instead of pooling the fractions with EVs, we actually measured each fraction individually and again measured the candidate protein in addition to the tetraspan in CD96381 and albumin. And when we did this for L1-CAM, which was a commonly used neuron marker that people had used, we found that both in CSF on the left and plasma on the right, L1-CAM elutes in the later fractions very similarly to albumin. And this was surprising because many people were using this for pull down and detecting things after the pull down. And if we use density gradient just to confirm with another method, again, we see similar results where the L1-CAM is in the same fractions as albumin by density gradient and not in the fractions where we see the tetraspanins, where the EVs are by Simoa. And so we think L1-CAM is actually a free protein that's not associated with EVs in plasma and CSF, even though canonically on neurons, L1-CAM is a transmembrane protein. And there are actually reports of L1-CAM being cleaved in different scenarios, which could be why it's a free protein, as well as a report of alternative splicing where just the transmembrane domain is actually taken out with alternative splicing and again, generating a free protein. So I think L1-CAM is not the right cell type specific marker for neurons, but we wanted to ask more generally, what is a cell type specific EV marker and come up with a framework for any cell type to pick the best candidate marker. And the framework is very simple, and we have a preprint describing the details if you're interested. But the idea is we're taking 3 criteria and we're applying them to all proteins to see which ones should be prioritized. So we want things that have a transmembrane domain so that we could pull on them with an antibody and get the EVs. We want things that are cell type specific at the gene expression level. And then we want things that are also present in EVs. And for that, we use EV proteomics using mass spec, which doesn't depend on antibodies, that we generated for CSF and plasma using the multimodal resin method that I described earlier with Captacor beads. And so if we do this and plug in the real numbers for neurons, we get 28 candidates. Of course, this number would change depending on how exactly you pick your parameters. And then these are the 28 candidates that we detected in either CSF or plasma and are neuron and brain specific by bulk RNA-seq. We also looked at single cell RNA-seq data sets, and the ones in bold are also specific to neurons by single cell RNA-seq. And then we started making Simoa immunoassays to each of them. And many of them were not found on EVs or not found in the biofluid or not found in the EV fractions, but we found one candidate, Nerexin-3, which is on EVs by SEC. So again, similar to what we did with L1-CAM, we took the individual fractions of CSF or plasma run by SEC using Cephero CL6B. And then in each fraction, we measured CD9, albumin, and Nerexin. And you could see that Nerexin is in the early fractions of both CSF and plasma, like CD9. And that's the only candidate so far that we've been able to see of those 28. We couldn't make immunoassays to all 28 because many of them don't have antibodies. But of the ones we tried, that was the one that we could detect the EV fractions. And then again, we repeated density gradient as an orthogonal technique, and found that in plasma, Nerexin is also on EVs by the density gradient centrifugation, with then using Simoa to measure each fraction, like I mentioned before. So the details of that are on Bioarchive. But the next steps that we're actively pursuing is trying to characterize these Nerexin EVs and look at their cargo using a variety of different methods, using single EV imaging, using various methods to look at the RNA, both using qPCR for particular transcripts as well as RNA-seq to try to get all the transcripts. We're looking at specific cargo proteins using Simoa, so internal cargo proteins, including trying to confirm the neuronal nature of these EVs by looking at whether there's enrichment of neuronal cargo proteins and disease-associated proteins like alpha-synuclein for Parkinson's or tau for Alzheimer's, and also doing mass spec to characterize the protein content in an unbiased way of these EVs. Now, one of the really big challenges with doing cell type specific EV work is in many cases you're really getting a very small subset of the total EVs. And what that means is you're faced with many challenges from non-specific bindings. So if you use magnetic beads conjugated with antibodies, you're always going to get some non-specific binding of proteins to your bead. And so in some cases, you can get less of this binding by first purifying the EVs with SEC. And so we kind of compared that in the preprint and showed that we can, with a good antibody, you can do a pull down without that, but it definitely reduces the background if you first do SEC. But even if you do SEC or some other method, you're still going to get background, both from non-specific binding of EVs without your protein, as well as free proteins and lipoproteins. And so we always do an isotype control non-specific antibody for any pull-down experiment. And in many cases, when we see something with the pull-down for Nerexin, we found that you often can see that with a non-specific antibody as well. So we're spending a lot of time really figuring out how to optimize every step of this pull-down and how to get rid of background as much as possible. So again, this is something that is much more challenging than the total EVs, where, as I've shown you, we think that works really well. But of course, still very exciting to try to get cell type specific information, which is a unique advantage potentially of EVs. And so I want to end now to make sure we have a lot of time for questions. But just to summarize what I told you, we're thinking about EV isolation from the perspective of we have to have a way to measure EVs quantitatively. And because particle methods don't measure EVs quantitatively, we think immunoassays to measure EV proteins are the way to do this. We use Simoa, but for many things, you don't necessarily need the sensitivity of Simoa. For plasma, you might have for a lot of biofluids enough sensitivity with just ELISA. With the assays we had, we optimized SEC after comparing it to other methods and found that the resin that gave us the highest yield was Cephros CL6B, which makes sense because it has a smaller pore size than 4B and 2B. And so we use that for the majority of our applications where we care about high yield. But as I mentioned, you can also think about combining this resin with other resins and make it a high purity isolation with resins such as Capticor, which can be used as a slurry after or in a bottom layer with Cephros. The main challenge with SEC was EV isolation, and we think that this challenge can be solved by using a plate-based SEC where you have different wells and each well is equivalent to a column. And if you have this 24-well plate-based SEC, it can be automated such that it can actually scale to hundreds or potentially thousands of samples per instrument per day. And so I talked about this multimodal resin method that can be used in a slurry or in column. We're using that particularly for mass spec. And then I also mentioned that we have a lot of work going on cell type specific EVs, including a computational pipeline that's available for anyone who's interested in the preprint, and on GitHub we have the code for it. The links in the preprint for finding candidate markers for cell type specific EVs. We're focused on neurons in the brain, but this can be applied to other cells of the brain or other cells outside of the brain. And then we're focused on trying to validate the marker that we found is on EVs, that's cell type specific Nerexin-3, and really working on the challenges of getting enough signal to background with this pull down using both RNA and protein. So that's what we're doing, and I think we have quite a bit of time for questions, so I'm excited to have a bit of a discussion based on what I said.
Great, thank you, Dima, for the great talk. I have a few questions already queued up here. The first one is, what's the sample size that you can put on those 24-well plate for ICC?
Yeah, so we basically tried to make the 24-well plate analogous to the column. And for the column, we usually use 0.5 ml or 1 ml. We also can use less. So we have done it with 100 microliters. So the volume is quite flexible. You just might have to, if you go over 1 ml, you'll have to adjust where the elution is. And you might get less purity just because you need enough resin to separate the sample. If you go less, we usually don't like to use for any kind of biomarker application less than 100 microliters, just because then you're getting a lot less material, and EVs are still low abundance for most things that you want to detect. So usually 0.5 ml to 1 ml is what we use, but theoretically you can use anything less than 1 ml and even more than 1 ml. We haven't tested how high you can go, but certainly 2 mls would still work.
Another question I had is, have you tried ultrafiltration for purifying your EVs after sec? And more specifically, have you compared 10K to 100K cutoff?
Yes. So Jan van Duyn had a really nice paper where he compared lots of different filters. Jan van Duyn and his collaborators, he developed the dual mode chromatography. So he had a paper, this was from Ann Hendricks' lab at the time. We can put it in the chat. And so I'll find it and put it in the chat in a minute. So this was a paper where they compared a lot of different filters, and they found that the best were the Amicon 10 kilodalton filters. And for some reason, the yield was higher than the Amicon 100 kilodalton filters. So that's what we've been using, and we confirmed that those are quite high recovery. And
there's a question about size. So there's a question saying EV isolation. Have you tried looking at the size distribution of EVs isolated by different resins? They're most specifically caring about, it says SC tends to recover smaller EV subtypes. And what would you recommend for recovering larger EVs that are larger than 500 nanometers?
Yeah, so we try to think about EVs from the perspective of biomarkers, where we don't know whether smaller or larger EVs are better. So we try to get all of the EVs together. But certainly you will be able to get, if you take the earlier fractions, you'll get larger EVs, and the slightly later fractions, you'll get smaller EVs. And you could specifically optimize that. For that, you might not want Sapphiros CL6B, you might want a larger pore size. But the challenge there is to have a really good technique to actually measure the size after you change those parameters, because as I mentioned, with size quantification techniques, often they're not accurate, and often they can discriminate lipoproteins from EVs. So that's something you could do, but you just have those technical challenges. And for our purposes, we have not done that. So we use a 0.4 micron filter just to be very sure we don't have any cells or platelets. You don't have to do that, but we do. And then we get all of the EVs below that go through the filter.
Awesome. I have another question about proteomics. So I think there's two questions I'm combining. Is basically essentially they're saying that SEC dilutes you dilutes your sample, you get a larger volume at the end. How do you make sure you have enough to run proteomics?
Yeah, good question. So I think we do use filtration. So we use these Amicon 10 kilodalton filters. Again, I'll put the paper that did the comparison from another group, from Ann Hendricks' group, in the chat in a minute. But in our paper, we detail how we do it, and it's basically using these filters afterwards, because for most applications, you do want to get your EVs into a small volume to increase your sensitivity, including for proteomics. And that is what we do. I just found the paper and I'll put it in the chat right now. So it's this paper that compares a lot of different filters.
So there's a few questions here from Paulina. I'll try to summarize them. Basically, it's more about the biogenesis and about the brain EVs. The question is, do you know anything about the transport mechanism of EVs from the brain into the bloodstream? And the second part really about, do you think EVs fuse with each other and aggregate in plasma?
Yeah, so we don't know much about the transport of the brain, of EVs from the brain. So, there are some people who have done some mouse experiments where they try to either inject them into the brain with a tracer of some sort and then see if they come out, or to make some transgenic neuron-specific promoter. All of those experiments, I think, have limitations, even though they're interesting. So I think at the end of the day, we don't know. And what we have just tried to do is pick the most neuron-specific genes that, to the best of our knowledge, looking across all gene expression data sets, are only expressed in neurons. Of course, whether that's 100% the case or not, you could never be sure. And then prioritize those and see if we can detect them. And as I mentioned, Neurexin 3, we can detect, and it's one of the most neuron-specific genes that we know of. And then the second question was about EV fusion. Yeah, that's also one that's actually quite difficult to prove or disprove. Some people have looked at that. I think the general notion is that they don't fuse because there's a fair amount of of energy required to make vesicles fuse, and then they also have proteins on them. So I think some people have looked at that in certain experiments. I think it's hard to unequivocally prove or disprove, but I think the general studies that have looked at it, the assumption is that they generally don't fuse after they're released.
There's a lot coming here. One person asking, can you share what a Nexin 3 antibody you used?
Yes. So for the Neurexin 3 antibody, we actually have tested every single commercially available antibody, and all of that information is in the preprint, and the one that worked the best is described in the preprint. So you can see all of that data. We did a lot of different experiments to compare different antibodies. So all of that information is in the preprint.
So there's a question here about cell-specific EVs. So how do you believe there's enough circulating cell-specific EVs in plasma to yield enough data from proteomics?
Yeah, that's a great question. The answer is we don't know. So we can pull on Neurexin and get proteomics data, especially now that we're using, so there have been developments in mass spec technology. So for example, there's a new instrument that came out last year called the Astral that we just got access to, and we can measure things, but the challenge is not can we measure enough, because we can. The challenge is, can we be truly sure that it's not non-specific binding? And we're doing a lot of different methods that we're trying to develop to prevent as much non-specific binding, including, for example, ways of releasing the antibody from the beads before analyzing it to prevent the non-specific things on the beads. But the answer is we hope so, but we don't know. We haven't been able to convince ourselves yet that we truly are measuring the contents of neuronal EVs. So that is the main thing we're working on with that Neurexin aspect of the project.
For plasma EVs, how much yield decrease in sec versus sec plus resin? I'm assuming sec plus resin, the person is meaning sec plus the capillary slurry.
Yep. So all of that information is also, we described the multimodal resin method in our preprint, the same one with the neuronal EVs, because we used it for the proteomics of the total EVs. So we describe all of that there. But the short answer is with SEC, we get about 100-fold, and again, you can change that number to 1,000-fold depending on what fractions you take. You can not take the last fraction that we normally take. So let's say 100-fold or 100 to 1,000-fold with SEC. And then, as I mentioned, with the multimodal resin afterwards, we can get an extra 100-fold purity on top of that. So that's about 5 orders of magnitude of purity of EVs relative to albumin. But of course, if you do the multimodal resin, there is also loss in yield. And so we quantify all of that in the preprint in the supplement.
So there's a question here showing that you showed that basically L1-CAM is in the soluble, there's a soluble form of L1-CAM, but do you have evidence that L1-CAM is not also on neuronal EVs?
Yeah. So it's hard to 100% disprove that. What we can say is that a large majority, we think more than 99% of L1-CAM is in the free protein fractions. Because SEC is not 100%, right? You still get, when you do SEC regarding of which method, you'll still get some albumin because albumin is so much more abundant than other things. So, you will get some free proteins in your early fractions. And so it's always a question of if you detect L1-CAM above your limit of detection in your early fractions, which we do, is that just free L1-CAM that we can't fully separate by SEC because it's not 100%, or is that truly L1-CAM on EVs? And so we can't be 100% certain that it's one and not the other, but I think that most of it is still free L1-CAM because we're using a very sensitive Simoa immunoassay, and if it's just barely there by that, I think it's very, very unlikely that there's going to be enough neuron EVs to measure cargo inside of those. Again, I can't disprove 100% that there's no neuron EVs, but it's just the question of, if you are doing this and you have precious samples, how are you going to prioritize which marker you're going to use? And for me, L1-CAM would be a very low priority, but we're comparing the markers that we found, including Nerexin and others that have been described more recently, to L1-CAM all against each other to basically see from the same pool of plasma or CSF, how do they directly compare? And at the end of the day, that'll be the most useful comparison for choosing, I think. But I think we wanted to select something that we could detect clearly on EVs, which was, again, Nerexin 3 and not L1-CAM.
Thank you, Dima. So there's one question I believe I can answer. So someone said they've used the apex column and they, sorry, there's a lot of questions. I'm trying to find it again. So they've used the apex column and they see high EV recovery and they see EVs in fractions 1 to 3. Can they use only one fraction 1, 2, or 3 for mass spec? The answer is yes. So you can pull those 3 fractions and concentrate them, or if you think there might be different EV's properties in the different fractions, then yeah, you can run fraction 1, 2, or 3. So we've done that actually repeatedly to see if we see any biases in the different fractions. So another question here to Dima is, can I use Capricorn beads without the size exclusion column?
Yeah, so that's described in the preprint. And as I mentioned, for cerebrospinal fluid, which has about 100-fold less protein than plasma, you can use a mil of CSF with no SEC and the Capricorn beads. But for plasma, if you want to use a mil you theoretically could, but you would need a huge amount of capture core beads. That would be very expensive and also just impractical. You'd need like a very large, it wouldn't be in a 2 ml tube. So if you wanted a very small amount of plasma, like 10 microliters, 5 microliters, then yes. But if you wanted a larger volume of plasma, which is generally what you want, like 0.5 ml or mil, we found that the capacity of the capture core beads is too low, and you first have to use SEC before.
Perfect. So there's a few questions asking about, is the neural specific EV isolation kit commercially available? So we work closely with Dima and Dima's group at the Wyss. So that's something we potentially will make commercially available once we've gone through full validation and sourced the materials to be able to commercially make it available. So there's a lot of processes that we need to go through to make something that you can run in a lab, in your own lab, versus make it reproducible to be run in everyone else's lab. So that's something we'll be working on. Currently what we have is to do some of these screenings. So there are magnetic beads available currently that you can leverage, but also you can also use our ELISA kits that allow you to have custom capture. So if you're interested to know, does my marker X is on EVs, and if these EVs you believe have tetraspanins, you can leverage our ELISA where you can capture with your marker X and detect with the tetraspanin, so you know you have co-localization and that marker is present. I think I've covered all the questions. If I have not, please reach out to us. I'll have our contact information on the screen here shortly. We're happy to answer all your questions. So Dima's email's right here. So if any question about the talk that was not able to cover or missed, please feel free to reach out to Dima. If you have any questions to us at Everest, me and Tal is our head of research. We're happy to answer any of your scientific questions about the products. How do they work? You have any new ideas that you would like us to address in this space? We'd love to hear what you guys have to say. If you have any questions about our products, how to buy them, are they available where your lab is in your geography, please reach out to Dimitri and he'll be happy to help you guys. Again, really appreciate your attendance. We're happy that you were able to make it here, and have a great rest of your day.
Thank you. Bye, everyone.